Hello, I'm Janice Juraska, the PI on Project 3, and I want to talk to you about the results we have so far on perinatal BPA, the cortex, and behavior. We also have studies ongoing on the phthalates, but we don't have any results from them yet. I also will go back before this current P01 to the P20 studies because our current work is based on those. I don't have to tell this audience that BPA, bisphenol A, is everywhere. It's in plastics, it's in resins, we've all been exposed to it, the environment is full of it, and in addition, it leaches out of products. Now, a lot of people know intuitively, and it really is true, that prenatal development is particularly vulnerable to outside forces. In humans, all cortical neurons are born before birth. And the pruning of excess neurons, something that a lot of people don't know about, but there is pruning, is also started. In addition, there's evidence, both in humans and in rodents, and rodent models are what we're going to be using here, that estrogen receptors are present before birth. BPA binds to estrogen receptors, and so it is capable of disrupting the processes of producing neurons and pruning them. So we exposed rats perinatally to BPA over a time period that approximates human prenatal exposure. Rats are born more immaturely than humans, so we continued exposing them after birth. Okay, so here was the original P20 design. Long Evans hooded rats, I'm always using them. They're pigmented, they see, they're good for behavioral experiments. They are exposed to BPA from the beginning of gestation to the 10th postnatal day after birth. And we had two major endpoints that in adulthood, in other words, they were exposed early on and then they just grew up and we looked at them as adults. Actually, first was the cognitive behavior. I'll be describing that, a 17-arm radial maze. And next, we harvested their brains and looked at the number of neurons in the medial prefrontal cortex. Why did we look at the number of neurons? Neurons are now generated in the cerebral cortex after birth. So changes in neuron number can be permanent. This contrasts with a lot of other neural elements, dendritic branching, synapses, neurotransmitters, that can change in adulthood. So neurons are the most basic and the most permanent part of the neural elements. There also are sex differences that we have found in my lab in the loss of neurons in the cortex in rodents, we don't know about humans, during early development and in adolescence. And a lot of these losses, a lot of these sex differences and losses are influenced by gonadal hormones. Why did we look at the medial prefrontal cortex? The human prefrontal cortex is important for executive functions. And it is involved in just about every neural disease that has been described. Includes schizophrenia, depression, autism, and addiction. The medial prefrontal cortex is the rat analog of the human prefrontal cortex. Now, the first step in quantification of the number of neurons in the medial prefrontal cortex, for those of you who are really into this, it's the areas PL and IL, is to calculate the volume of this cortex in consecutive sections through the structure based on cytoarchitectonic characteristics that are illustrated here, but I have to admit that considerable training at the microscope is required for everybody to see where these borders are. However, the borders have to be constructed, and then we figure out the volume. Then this is a higher power picture of what was going on in the last one, so that you can actually see neurons. The big blue structures are neurons, and there's some little white arrows pointing to glia. Given that we're going to be counting neurons, we also counted glia. That's a separate story I don't have time to talk about, but it's an interesting one. So we did this with the optical dissector. It's a stereological technique. It is not influenced by the size of any given structure. In other words, it's not influenced by the size of the cells that you're counting. Once we do that, we get density, which is the number of neurons per volume, and we multiply that by the volume of the area, and that results in the number of neurons. It's very easy to get density and number mixed up. Density is just what's in a small volume. Number is how many are there totally. The number of neurons in the medial prefrontal cortex. And here we have two different layers doing basically the same thing, but there were significant differences in the lower layers, which make up the majority of neurons. And you can see that females had no differences across the different doses, right? No differences from 0, the controls, across the different doses that were used. But in males, at the highest dose, there was a significant increase in the number of neurons in the medial prefrontal cortex compared to controls. You can also see that there is a tendency, which was not significant, in the upper layers. This has been published recently. What is especially interesting about this is that this is in the direction that you see in young human autistics, male autistics. Not that many females have been looked at. And it may indicate, we don't think we have autistic rats here, it may indicate a tendency for an increase in neurons that may predispose an organism to autistic symptoms. Well, what about an advanced cognitive behavior? The 17-arm radial maze, baited and unbaited arms. This is a very complicated task. There's 17 arms. The rat is put in the central platform. There's food in all the baited arms, which are the gray arms, and there's no food every day in the unbaited arms. The rats have really two tasks here. One is to avoid the unbaited arms. We score those as errors, right? Because they're never going to get food there. And also to retrieve the food, which they're pretty cooperative about doing, retrieve the food in the baited arms, but only go into each baited arm once in a day. This is a very hard task. There's 17 arms. We run this for 4 weeks, 6 days a week, and rats do get better at it. So, once again, these were animals who received BPA early in development, then they grew up, and were doing this task. We did not find any interesting, significant differences in this task, amazingly enough. For the unbaited arms, right, which is the reference memory or long-term memory portion, you could see that females really show no differences. Males, it looks like at the end of the 4th week, that males controls look like they are doing better, fewer errors than the other, than anybody who was dosed, any of the males who were dosed. But this is not significant. All right. So even there was a block by treatment effect, but it was only marginal, so we did not go in and pursue it. Likewise, for the baited arms, females really didn't show anything. Once again, it did look like males who were controls did better than the BPA-exposed animals, but it was not significant. From this, we can conclude not that there's no effect of BPA, but that the effect of BPA, if there is one, on cognitive behavior is not profound. I will come back to that. Now, we're talking about the current center, the PO1 design. And once again, we're looking at perinatal BPA, and we've added high-fat diet because it's so common in our country and because it may augment some of the effects of BPA. We dropped our treatment is 0, 40, and 400 milligram per kilogram per day of BPA. We dropped one of the doses because this experiment has yet another variable, high-fat diets. We have 2 diets. The mothers are fed while pregnant and then for the first 10 postnatal days while their pups are being dosed. Either a high-fat diet, 45% fat, or a low-fat diet at 15.8% fat. Very few Americans have a low-fat diet at 15.8%. All right. Both treatment and diet are occurring during dosing. We're looking at both males and females. So if you calculate the groups, there are 12 groups here. Consequently, we're doing this in cohorts that take a while to finish. And you can see we have many, many endpoints, right? Here's our dosing, right? And then various measures are being taken, and I'm not going to be able to report on all of them, and in fact, we haven't analyzed all of them yet, but they are ongoing. This is just to impress you. All right. I am going to talk about attentional set shift here. This is another complicated task, and it's a measure of cognitive flexibility. It's dependent on the prefrontal cortex, and it's an extra-dimensional shift. The rats first learn to either discriminate as the positive place for food, either black arms or white arms. Once they reach criterion in this, it's not that difficult a task. They are switched to either a rough surface on the floor of the arm or a smooth surface. This is a shift in what the positive cue is, and it's a shift in the modality, and it is a hard task. Bethany, this was developed by these people, but Bethany Kunoud in my lab is presenting a poster on these results, and so I'm not going to talk about it here. Suffice it to say that it's yet another task that tells us that BPA is not having a profound effect on cognitive abilities. But it does seem to be having an effect on social behavior. We looked at a bunch of adolescent rats that had been dosed early, and they are housed after weaning with a same-sex cage mate. However, when we do this test, we isolate them for an hour so they don't have anybody around, and then we introduce them to a new rat. And we do this for 20 minutes, and we calculate every minute what each rat is doing. And for the first 2 days that this goes on, we do it for 4 days, all the groups are about the same. And they're a little hesitant to do a lot of social behaviors. By the way, the social behaviors are diverse. That's sniffing, chasing, and wrestling, which is a really common behavior in adolescents when they're playing. But by the last 2 days, the controls that hadn't been exposed to BPA are doing a lot more social behaviors, significantly more social behaviors than either of the doses. Once again, this is kind of an indication that early BPA may model some of the symptoms of autism. That is as far as we've gotten. We're running a lot of other tests, and we're looking at the anatomy, but I'd like to thank the whole crew of people who have worked on various aspects of this, and of course our funding.